بسم الله الرحمن الرحيم. موضوعنا PCR polymerase chain reaction, a full simplified explanation. Hello my students, hello everybody. Now we will study the most important technique used at recently years, the most technique and devices used especially at the last conditions of spread in coronaviruses. What is PCR? PCR is an exponentially progressing synthesis of the defined target DNA sequences in vitro. It was invented in 1983 by Dr. Mohamed, for which he received the Nobel Prize in Chemistry in 1993. Everyone, this video speak about the really interesting technique used in widely nowadays, which is called PCR polymerase chain reaction. I'm going to speak about the principles and the technique procedure. What is the PCR? It is a DNA amplification technique, generating million copies of a particular DNA sequence. This device is used to determine and amplify the sequence of a specific gene. We extract DNA from a human cell or any organism, and we are interested a particular gene or a particular sequence of DNA. So this device, it is so important. It can be used in several purposes. To obtain a million copies of a specific target sequence of any gene for any organism, you use this technique, PCR or polymerase chain reaction. As illustrated in a front figure, we want to amplify or to obtain multiple copies of a particular sequence of that DNA, which limited between two red lines. Okay, like that, from this to this is we want to increase the number of that target sequence. Okay, to know or to carry out this process, initially we want how does the technique work of PCR. This technique depends on which is called thermocycles, as illustrated in PCR graph. It is depends on the increase or up temperature and decrease this temperature in a few seconds. However, such biochemical reactions or PCR mixtures is completed in cycles, as showing in this figure, which reveals the relationship between temperature and temperature. This is increase or decrease, and the time of that thermocycle reaction. We noted that there is two cycles as illustrated in this figure, first cycle and second cycle. Both are selected to illustrate the mechanism of such reaction. The first one of cycles started from this point, starting point, and ended with the red line, and also the second started with the end of first and ended with the third one. Okay, we note that there is a up of temperature, then down. Okay, this is a first, and second also repeat this mechanism like that, up then down of temperatures of such bio reaction. These cycles led to increase the number of selected. We will study how does the technique work in PCR. This the most important technique. Okay, this is tool-led DNA generations and formation of new copies of DNA. It must be prepared this the smallest Emmendorf tube of PCR, and this is the most important or the general component of PCR technique. Firstly, we will add a template of DNA or parent DNA having the particular sequence of that gene we want to amplify it. Also, we added the DNA contents DNT base. DNT base are separated nucleotides or blocks, which from them we construct the new strands of that bioreaction. Having this DNT base having the four types of nucleotides, C, cytosine, G, guanine, A, adenine, T, thymine. All these components are in separately. Okay, so this diagram showing the connection between these nitrogenous bases A, T, G, C, and this is a structure of formula structure of DNA, deoxyribose sugar. This nitrogenous bases binding with the first carbon atom of deoxyribose sugar, and the phosphate group attached at the fifth carbon atom attached with the phosphate group, and this is the nitrogenous bases which attached with the first carbon atom. Okay, this is the main structure of DNTP. The third one which must be added to this biochemical reaction of PCR technique, primer. Primer having two types: forward primer and reverse primer. Forward primer, it is from 5th prime to 3rd prime, and the reverse from the 3rd prime to 5th prime. This primer are ranging about 20 nucleotides. It is a single short piece of DNA, and matching with a DNA, old DNA or template DNA, according to their sequence which prepared before this reaction. These primers well attached, so they design it to complementary by base pairing between the primer and strands of template DNA. From this primer, the elongation of DNA going toward, as illustrated by these arrows, going toward each other at opposite. This is directed for this direction, and this is directed to elongate the DNA at these directions. This from this shorter pieces of DNA at which the elongation of DNA continuously occur. Also, we added the DNA polymerase. This enzyme is a highly thermostable DNA building up enzyme. However, it is extracted from bacteria called Thermus aquaticus in about 1976. DNA is also it is most important, by which the tag polymerase take them to build new strands of DNA. This is represents the components of PCR mixtures like that. Firstly, we added template DNA containing the target sequence of this which desired in a specific gene. Oligonucleotides primers, as we say before, forward and reverse primers. Thirdly, with a tag polymerase as a DNA polymerase, which responsible for build up new strands of DNA in these conditions. Fourth, DNA base to build up new copies of DNA strands, including DATB, then DCTB and DGTB and DTTB, adenine, cytosine, guanine, and thymine. Also, we added a buffer to maintain pH and provide with this magnesium chloride. Magnesium chloride to enhance and induce the DNA amplification by activity of tag polymerase enzyme, which is used. Okay. We also added that deionized distilled water. Okay, and maybe covered with mineral oil, mineral oil, to prevent the evaporation. What is the process of PCR, or what is the procedure of PCR, and how does PCR work and its steps? Please take care that it is a thermocycle steps. Don't forget, it is a thermocycle steps, which repeated several times in each thermal cycle. So we have 3 steps of the temperature changes in each one cycle. Okay. The 3 steps of temperature changes started with high temperature. Started with a high temperature, then cooling to it, and then up gradually. So the first step is called denaturation of hydrogen bonds. As you know before, that the 2 strands of DNA bind between them by hydrogen bonds. Hydrogen bonds. Hydrogen bonds may be 2 hydrogen bonds between A and T, or 3 hydrogen bonds between C and G. You see that 2 red colors, meaning that we need to copy this specific sequence. Okay, so this is the particular sequence we need to amplify it. The hydrogen bonds between the 2 these strands which bind together between them, it must be need to break these hydrogen bonds between them, between 2 strands. So it is called denaturation of hydrogen bonds. The denaturation hydrogen bonds, like that, we cut the segment of DNA to explain how you can break between these hydrogen bonds between the bases, nitrogenous bases of nucleotides of DNA for both 2 strands. Okay, so you need the temperature about 94 to 98 Celsius, which take about from 20 to 30 seconds. This temperature up to PCR mixtures about 95 approximately for 20 to 30 seconds. Okay. After this time, the hydrogen bonds between the 2 strands will be broken. So I will have 2 separated strands, 2 separated strands of DNA. By the first step of denaturation of hydrogen bonds. This needs a time depends on, as we say before, the number of hydrogen bonds between C and G or between A and T. According to these bonds, the number of A T or CG in this segment, this temperature needs, or this step needs a time more or less according to the number of C and G or A and T in this segment. Now, the third step of PCR technique or thermocycle steps is annealing step. Annealing meaning that the DNA is recombinant by primer in a double strand form by hybridization between this target sequence of DNA and primer you used. Whereas this attachment between them, primer and DNA sequence or template DNA, is occurred by complementary binding via hydrogen bonds to form double-stranded polynucleotides. However, such step take a time about from 20 to 40 seconds at temperature of from 50 to 65 degree. At this figure, we need to perform such a step of annealing by adding two primers of forward and reverse one as starters for this bioreaction technique. Please take in your consideration that binding between them to building up new strands by tag polymerase, and this tag polymerase act from 5th prime to 3rd prime, which represented by two arrows, blue arrows in the up figure, and both primers act in opposite direction. Therefore, both primers act in opposite direction. This is so important point. Okay. To calculate the annealing temperature, use this equation. This equation of melting temperature. Melting temperature, abbreviated with Tm, as illustrated in the up figure. Okay. Melting temperature Tm of that primer depending on content of AT or CG nucleotides, which is found in the structure of designed primers, forward and reverse primers. Tm or melting temperature is defined as a temperature which necessary to dissociate the double helix of DNA into two single strands and indicate also the duplex stability of the new structure by hybridization between template DNA or target DNA and the primers. For example, when you need to calculate primer at and you haven't the annealing temperature for using of it in this bioreaction, it must be firstly calculate Tm of such primers, and in turn you use primer at annealing point lower than with a 5 degree. So if you calculated the Tm of this to any primers at 60, so the annealing temperature is calculated as 55. When Tm 60, the annealing temperature is lower than by 5 degree and became 55 degree, and so on. However, we will going to the third step of PCR protocol. The third thermocycle steps of PCR, it is called elongation step. After matching that both two primers, these primers need to expand it on the each one on the separate of DNA two strands. The addition of new nucleotide to build up complementary strand to the first and complementary strand to the second DNA strands at the position of hydroxyl group, at which the new nucleotides added to the primer. Okay, this is led to the extension of these strands, both strands, forming finally with two strands. For the first one and the second one, forming old DNA strands with a complementary with the new strands, and another one also forming one from old or the parent strands were attached to the new strand. This step consumed about 2 minutes at the temperatures from 75 to 80 Celsius degree. This step it is so important because you see giving the aim of this reaction or this technique. So it is called elongation step, forming two strands from the first strand when we start. Okay? At the end of elongation step, we will get two separated copies of DNA or two copies of a particular sequence as we need. At normal case, we need 1 minute to build up a new 1000 bases. However, look at this the thermocycle steps of PCR. You note that it started with the heating up temperature to about 95 Celsius degree. This step for DNA denaturation, as we told before. So that step of elongation or extension to separate between two DNA strands and to form two copies of the target gene or sequence. So after this step or denaturation, DNA denaturation, the cool down at a temperature about 50 Celsius will apply to hybridized DNA with two primers. Then this temperature up again to about 72 Celsius to carry out the elongation or extension step, which performed by the action of tag polymerase enzyme to build up new nucleotides at the direction from 5th to 3rd prime in both two new strands. The end of elongation is also the end of this first cycle, as you see in this figure, which lined with the red color. At the end of elongation, it is also the end of first cycle or thermocycle. So now we obtained 4 copies of a target or desired gene or sequence we determined before. So let's see if we started with the 1 copy of desired sequence. At the end of first cycle, the product is 2 copies of this sequence. Then after the second cycle, we get 4 copies of this desired gene, and in turn after the 3 cycles became 8 copies. So after 3 cycles of 1 copy of DNA sequence, it multiplied into 8 copies, and so on, so on. It's like a cascade. So, how many copies of DNA after the end of a certain cycle? I will give you an example. What you get if you run PCR for 1 hour? Since the one cycle takes around 2 to 3 minutes, and whereas if 1 hour equals 20 cycles, and therefore the number of copies will be 2 power 20. Okay, which gives more 1 million copies, as you see in this equation. Okay. This is according to the equation as illustrated in the above of figure, which showing the number of copies of DNA sequence equals 2 power n, whereas n expresses the number of thermocycle of PCR. So such a lot of copies it can use in cloning of sequence and other applications, as illustrated in the next slide. The summary of PCR protocol involves 3 scopes. Firstly, what is the PCR? It is a thermal cycler and used to amplify a specific sequence of DNA. The second scope is PCR components, which have the following: DNA sample, primers, nucleotides, DNA base, tag polymerase as the DNA building up enzyme for DNA, mixed buffer containing magnesium chloride. All of this put into a PCR tube. The third one scope of PCR includes the PCR process as a thermal cycles. Each one cycle consists of 3 stages: denaturing, annealing, extension. Each one depends on the temperature. First, at about 95 Celsius degree. The second stage, or annealing, which includes the attachment or matching primers with the template DNA at the temperature of 55 Celsius degree. And finally, the third one, extension stage, at the 72 for synthesis a new strand. The final step of PCR protocol is agarose gel electrophoresis for PCR run, in which the final product is represented with the dark photo and the DNA taking light appearance. Each column called lane 1, 2, 3, 4, and so on. Often we use and put the in the lane 1 the marker, DNA marker. This marker is expressed by base pair PB, and to compare between the final products of PCR for a specific sequence, which it can be determined also by its molecular weight in base pair. There are many types of PCR, such as single PCR, nested PCR, multiplex PCR, asymmetric PCR, quantitative PCR, and reverse transcriptase real-time PCR, which use to diagnose of coronavirus COVID-19. Now we reach to the most important step, which is called applications of PCR. As you see here, these applications involves several uses of this important device. PCR technique, it may be used in molecular identification, sequencing, genetic engineering. From these uses of molecular identification, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens. Pathogens at which this includes several and so important points. Pathogens of microbes, bacteria, viruses, such as coronaviruses, all strains, HIV causing AIDS, and HAV, HBV, and HCV, which causes hepatitis, viral hepatitis. So all these pathogens, it can be detected by this technique. However, there is another using for the PCR technique as a sequencing, such as a bioinformatics, genomic cloning, forming several copies of target gene, and survey of the genome of a certain organism. Besides human genome projects, which has a lot useful usage in our lives. And finally, with genetic engineering, such as site-directed mutagenesis, gene expression studies. So this limited lecture showing the most important of this technique in our life, especially at last years. There is a lot information about PCR, but this is a summary for it, which it includes structure and applications, how to use of this technique. If you have any questions about this lecture, please leave your comment below this video. Thank you for your listening and your attention, and good luck for everyone.